The topic of this lecture is the molecular biological methods that are used at various stages in the drug discovery and development process. The aims of the lecture are to explain how molecular biology is exploited during drug discovery and development. Firstly, it's used to identify the genetic causes or drivers of disease. Secondly, it's used to identify novel drug targets. I gave one example of this in Friday's lecture. And finally, molecular techniques are used to validate drug targets. I will introduce you to a new concept, the orphan receptor, and to an experimental approach known as reverse pharmacology to study orphan receptors. I will explain how molecular techniques can be used to create assays using reporter genes. Transgenic animals are a key technology for validating drug targets, so we will cover the basic concepts of how genes can be knocked down or overexpressed in vivo. The lecture will end with an overview of biopharmaceuticals and how they are produced. The take-home messages from the lecture and what we would like you to develop a good understanding of are as follows. The methods used to investigate the link between genes and disease and identifying new drug targets. Orphan receptors and how to identify them. The molecular techniques that are used to develop assays for use in compound screening. Transgenic animals and how they are made. And how recombinant techniques are being used to generate protein drugs. Genes can affect disease in many ways. The most obvious and straightforward situation is where a mutation in a single gene directly causes disease. This type of disease is known as monogenic, and it includes some severe diseases such as cystic fibrosis and Huntington's disease. Some gene mutations might predispose to disease, but the disease is only manifest if additional genes are affected. When a disease is genetically determined, but requires the combined effect of mutations in two or more genes, it is known as a polygenic disease. But most disease is more complex than that, being influenced by multiple genes, as well as the environment, including lifestyle. These are actually the most common diseases, and they can have the biggest effect on the world's population, including diabetes, cancer, hypertension, rheumatoid arthritis, and atherosclerosis. Knowledge of gene expression can help provide insight into normal physiological pathways, and identifying altered expression can help to build a picture of pathological processes, even where the diseases are very complex. Genetic association studies investigate genes in populations, looking for correlations between the presence of disease and variations in gene sequence. Differences might be found in individual genes, which may have a causal role in disease, say a monogenic or polygenic disease. Alternatively, regions of a genome may show variations linked with disease. The most common variation found is a single nucleotide polymorphism, or SNP. That is a person-to-person variation in one of the three nucleotides that encode an amino acid. In this example, the SNP would change the codes on each of the DNA strands from glycine and proline to aspartic acid and leucine. But because there are several codes for each amino acid, SNPs don't always have an effect on the amino acid composition of a protein. For example, if the third base in the upper strand were mutated to an A instead of the second base, glycine and proline would still be produced. SNPs are the most common type of genetic variation, affecting 1 in every 300 nucleotides. If particular alleles show a higher than usual frequency of SNPs in association with a particular disease, then they would be suspected of increasing the risk of disease. Finding the gene responsible for a monogenic disease involves studies that compare the genes of healthy and affected members of families displaying the disease. It is easier if the families affected are large, or there is a large number of families affected. Members of individual families have similar DNA fingerprints. You are looking for sequences that differ consistently between the healthy and affected members. It is much more difficult to define the genes involved in multifactorial diseases. Very large populations are needed in order to identify consistent links over and above the normal genetic variation among individuals, which has no effect on disease. This means that projects are often large, not carried out by one drug company or a single scientist. For some of the big diseases, like cancer and cardiovascular disease, there are nationally organised projects involving hundreds of scientists. An example of this is the UK Biobank, which was set up to study samples of the UK population over time. It is in the early stages, but a range of data is continually being collected, from simple measurements like weight and blood pressure to blood and DNA samples, as well as brain, heart and bone scans. The two main technologies used for analysing gene sequences are DNA arrays and bioinformatics. Gene arrays consist of a small glass plate encased in plastic. The surface of the plate is covered in thousands of short, synthetic, single-stranded DNA sequences, which together make up the normal genes being studied. These arrays generate lots of data, requiring complex statistical analysis. There are now online searchable databases in which individual scientists store their results, making them available for other scientists to view. In this way, a global picture is being built up of all possible variations in the human genome associated with disease. Anyone can view the data, including you. An average gene array may contain 35,000 genes, which allows a large number of genes to be analysed in a single experiment. The general approach is similar for gene expression profiling and identifying SNPs. In each case, sample DNA is labelled with a fluorescent dye and inserted into the chip. Any sample DNA that is complementary to the chip DNA will bind to it or hybridise. The fluorescence is then detected, with the level and location of the fluorescence giving information about the genes from the tissue samples. In the case of expression profiling, RNA is extracted from tissues. It is converted into cDNA with reverse transcriptase and amplified using PCR before being applied to the chip. The readout from the chip is the level of fluorescence associated with each gene. The stronger the signal, the more expression. For identifying SNPs, the starting material is genomic DNA, which is denatured to give single-stranded DNA. Since only the normal sequence will be complementary to the gene sequence on the chip, the SNP can't bind, and its presence will be seen as a lack of fluorescence where the gene should be. This slide shows a fluorescence image of a portion of a treated gene array. A mixture of two samples of cDNA was applied. One sample was labelled with a green fluorescent dye, the other with a red fluorescent dye. The colours you see represent the differential signals from the two samples. So where there is a green spot, the cDNA in sample 1 has hybridised preferentially. So that sample contains a greater amount of the complementary DNA sequence. Where you see a red spot, that sequence is more highly expressed in sample 2. And yellow spots show equal hybridization of cDNA from both samples. Image analysis software can automatically analyse the signals from every spot and report the relative expression profiles for the samples for each gene. But you need to beware. This is just a first step in looking for a target. Altered gene expression in diseased tissue does not necessarily translate into altered protein expression, and it cannot tell you if the altered profile is the cause of disease or a consequence of it. So this type of genomic study needs to be followed up with data from other types of experiment that test the effect of the gene on function. These studies include proteomics to determine if protein expression or function is altered in disease, as well as physiological and pharmacological studies which test for the functional effects of interfering with the gene products. Sequencing of the human genome led to a major advance in drug discovery methods. Knowledge of the gene sequences meant that, for the first time, drug screening programs had access to any potential human target. Cells can be transfected with the DNA coding for the target. The DNA undergoes transcription and translation into the human target protein, and a cell line can be established which provides an endless supply of the human target. The cell line can be used to set up an assay to measure the activity of the target. This makes it easy to screen the target for drugs that interact with it. The big advantage over earlier methods, which depended on animal cells and tissues, is that molecules interacting with the human form of the target can be identified early. This should increase the chances of them succeeding further down the line. Before the Human Genome Project began, many receptors were known. They were identified as a consequence of studying the effects of ligands on tissues and working out what they acted on. Receptors were usually named after the ligands that identified them. The ligands were often endogenous to the tissue, such as adrenaline and acetylcholine, which gave rise to the identification of adrenergic and cholinergic receptors. Sometimes receptors were named after exogenous ligands or drugs. An example of this is the nicotinic cholinergic receptor, which was identified as the molecule through which nicotine produced its effects. Once the DNA sequences encoding the known G-protein coupled receptors were identified, studies of the remaining genome began to identify sequences with high homology to known receptor genes. But they were just sequences with no known function. They were presumed to code for unknown receptors, so were called orphan receptors. On the basis of structural homology, the human genome was predicted to contain about 800 unknown G-protein coupled receptors, or orphan receptors. So, how do you test if these genes do express receptors? And if so, what receptors are they? Or in other words, what are the receptors for? If you know the gene sequence, you can express it in a cell line and hope it will express a functional protein. But if it's a receptor, it will need an agonist to activate it, such as 5-HT to activate 5-HT or serotonin receptors. And you don't know what that is. So you start by throwing all the G-protein coupled receptor agonists you know of at it. But if there is no response, another approach is needed. It essentially involves fishing for ligands. Extracts are made of tissues in which the gene is expressed. The extracts are tested for their ability to produce a response, and gradually they're purified until the active ingredient is all that's left. The first successful classification of a receptor by this route was the ORL1 receptor and its agonist nociceptin. Being completely opposite to the classical pharmacological approach, this new gene-based strategy became known as reverse pharmacology. Another technology that grew out of the Human Genome Project is reporter gene assays. Reporter genes are gene sequences that code for a protein with a property that allows it to be easily detected. A length of DNA containing the reporter gene and a promoter region is incorporated into a cell line. When the promoter is present, it stimulates the transcription of the reporter gene into mRNA, and translation into the reporter protein follows. Two reporter genes can be employed simultaneously, provided they can be separately detected. This slide illustrates a typical cell assay employing a reporter gene to detect activation of a G-protein coupled receptor. In this case, when a ligand binds to and activates the receptor, the GS protein is stimulated, leading to activation of adenylate cyclase and production of the second messenger, cyclic AMP. Cyclic AMP in turn activates protein kinase A, which has many targets in a cell, including the Cre binding protein CREB. Upon activation, CREB translocates to the nucleus where it binds to Cre, the cyclic AMP response element, which is part of a DNA sequence just upstream of the luciferase promoter, which is in turn activated. This promotes transcription of the luciferase gene, resulting in the production of luciferase protein. Luciferase generates a luminescent signal which is detected optically and gives a direct readout of receptor activity. Reporter gene assays are widely used in drug discovery because they produce signals that are easily measured. And because cells don't normally produce this type of molecule, there is no risk of mistaking the signal. Because the reporter protein is synthesized only when the target is stimulated, the signal is very stable and reliable. The example on the last slide used luciferase as the reporter, but many other reporters are available, including a range of bioluminescent and fluorescent proteins. Bacterial beta-galactosidase is also commonly used because it can be measured using a cheap colorimetric assay. When X-gal is added to the cells, the enzyme converts it to an easily detected deep blue compound. This variety of reporter properties makes the simultaneous use of two reporters very practical. Transgenic animal models are animals in which the germline has been altered so that it either stops expressing the gene, overexpresses it, or expresses an altered gene. These are known as gene knockouts and knock-ins. A technology known as Cre-Lox recombination provides a method of targeting the DNA modification to a particular cell type, or triggering the modification with an external stimulus, so that it can be delayed until the animal is fully developed. This is known as conditional knockout or knock-in. You don't need to know the details of this process. All of these models are used to investigate how changing gene expression affects physiology and disease progression. They are also all used as models of disease for target validation and preclinical drug testing. Transgenic models are almost always mouse due to ease of breeding, fast development, and history of success in genetically engineering mice. This slide outlines the basic procedure for generating a transgenic animal. It starts with stem cells taken from an embryo, which are transformed with the DNA construct that will alter gene expression. The transformed stem cells are then inserted into an early embryo, which is implanted into the mother's uterus where it develops. The aim is for the modified DNA in the stem cells to enter the germline. Another method often used is to microinject DNA into the male pronucleus of the zygote. Once the pups are born, selective breeding techniques are used to generate the genetically modified animal line, usually a mouse line. Another type of transgenic animal is a clone. I am sure you all know about the first cloned animal, Dolly the sheep, which made headline news in 1996. Cloning involves removing the nucleus of a somatic cell and transplanting it into an unfertilized oocyte, which has had its nucleus removed. An electric shock then stimulates the hybrid cell to divide, and when it reaches the blastocyte stage, it is implanted into a surrogate mother. CRISPR is the latest form of gene editing and widely thought of as a revolutionary technology because it makes gene editing cheap and simple. CRISPR is short for clustered, regularly spaced, palindromic repeats, and it refers to a uniquely organised DNA sequence found in prokaryotes, which comprises short DNA repeats separated by spacers. It is key to the prokaryotic immune system, recognising and destroying foreign genetic material, for example from invading viruses. The mechanism employed by prokaryotes has now been developed for editing mammalian genes. It involves synthesizing short RNA molecules that match a specific DNA sequence, and these form guide RNAs that shuttle the molecular machinery of the cell to the intended DNA target in the genome, including the enzyme Cas9, which cleaves DNA. Once there, the molecular machinery can silence a gene or change its sequence. The technique involves a simple injection into oocytes with no need for embryonic stem cells. The final topic of the lecture is biopharmaceuticals, or drugs that are produced by a biological source. This description covers vaccines and protein drugs that have historically been derived from animals, such as insulin. But what I want to focus on is a new group of biopharmaceuticals known as biologics. These are recombinant human proteins. Clearly, in treating human disease, it is better to use human proteins than animal proteins. But proteins isolated from one human and given to another runs the risk of transferring infection. This used to be a major problem for haemophiliacs who could be treated only by clotting factors extracted from human blood. The introduction of genomic technologies has changed the landscape by enabling the production of recombinant human proteins on an industrial scale. The synthesis of recombinant proteins is directed by recombinant DNA, introduced into a foreign cell that has the machinery to transcribe and translate it into protein. Examples of recombinant human protein drugs available today include insulin and clotting factors, growth hormone and erythropoietin to stimulate red blood cell production, as well as tissue plasminogen activator to dissolve blood clots, for example in stroke. The list also includes humanized monoclonal antibodies, which act as antagonists in biochemical pathways. Etanercept, infliximab and trastuzumab are examples of monoclonal antibodies produced by recombinant technology. So what biological systems are used to generate biologics? The first drugs approved were made by genetically engineered microbes, usually E. coli, but yeast can also be used. But bacteria and yeast are not always able to reproduce the post-translational modifications of proteins that are important for activity. They may also fold the protein incorrectly, so recombinant proteins made using microbes can be lacking in activity. So production of recombinant proteins has largely moved to mammalian cell lines, mainly to Chinese hamster ovary cells. But these cell lines generate quite small amounts of protein, and extracting the protein with a high level of purity can be difficult and costly. An alternative source is transgenic animals. It may not be immediately obvious to you how that would be practical, but goats, sheep and cows produce copious amounts of milk, and the farming of these animals for milk is well developed. So the idea is to localize the expression of the recombinant protein in the mammary gland of one of these animals and make use of the natural milk-producing machinery to express the protein directly into milk. In 2006, antithrombin was the first recombinant protein from goat milk to receive approval from the European Medicines Authority. It is used for patients that have an inherited deficiency of the protein. It was followed in 2014 by Ruconest from the milk of genetically modified rabbits. And many other drugs are likely to follow in the near future.